Good afternoon, everyone. Good afternoon. If you can hear me, signify. Good afternoon. Can you hear me? If you can hear me, drop where you're listening from in the chat box. All right. Good afternoon. Good evening from anywhere you're hearing us from. I know you're all excited to be attending day 2 of the fully funded bootcamp. You're all welcome. You're all welcome. You're all welcome. I can see your comments in the chat box. Ayomide Okiti from Nigeria. Kola Sunny from Abuja. You're welcome. Gladys Honor from Lagos, Nigeria. Yes. Golden Selu Sandala from Zambia. You're welcome. You're welcome. You're welcome. I'm sure you all enjoyed yesterday's session, and I'm sure you're all ready to learn more today. It's going to be a complete practical session. So get ready. And I'm sure some of you already have junior software. Today is going to be an amazing. You're welcome. Nora from India. You're welcome. So if you have friends that are yet to join this program, kindly share it to them so they can also benefit from this program. I can see some people that joined yesterday are also here again. I'm so glad you're all interested in learning bioinformatics. You're all welcome. Kindly share to your friends and to your colleagues so they can also benefit from this program. It's one in a lifetime. Edith from Cameroon, you're welcome. Share with your colleagues and friends. Tanko, can you hear now? Henry from Nigeria, you're welcome. You're all welcome. The class is about to start. Kindly share with your friends, your colleagues, so they can benefit from this program. Good afternoon, everyone. You're welcome. You're all welcome. So in the next few minutes, we'll be having the CEO of Genomic to come and take the session personally. Everyone that will be joining this session is so lucky to be able to learn from such a man like him, the founder of, he knows a lot about bioinformatics. So get your pen and your notes and be ready to jot things down. You're all welcome. Share this to your friends so they can join in before the class starts. Yes, somebody's asking me today the last session. Yes, you can't afford to miss this. We have Ambrose from Cote d'Ivoire. You're welcome. Welcome. Kasar from Pakistan. You're welcome. You're all welcome. People from all around the globe, from Pakistan, from Nigeria, from India, from Zimbabwe, from Tanzania. I know you will definitely enjoy this session because the man, the individual, is the CEO of Genomic himself. He has taken it upon himself to find time and teach people this fully funded program. You're all welcome. Keep on sharing to your friends and colleagues. So now we welcome the CEO of Genomic Holdings, Mr. Oluwaseyi, to begin the session for day 2 of the fully funded bootcamp program on bioinformatics and genomics. You're welcome, sir. Is it really learning? God likes this one. The CEO will be joining us soon. There's a technical problem with the network. Okay, the CEO is here. You're welcome, sir. So I welcome the CEO of Genomic Holding, Mr. Oluwaseyi. He will be the one to take us through the day 2 session of the fully funded bootcamp in genomics and bioinformatics. You're welcome, sir.
Amazing, amazing. Thank you so much. It's amazing having you guys here. So we're going to be having quite a number of different hands-on training sessions. So we're just going to have a view of what we did yesterday, and we'll take it from there while we dive into the practical sessions. So just give me a minute.
So yeah, how about so please send to your friends and family while the CEO sort some things. You're welcome. Afik from Sudan, you're welcome. Uzai from Pakistan, you're welcome. Ogunwa from Nigeria, you're welcome.
All right, so thank you so much. Please, you can quickly invite your friends and family. Just remind them to join us right away at this session. And don't forget, I said today is going to be really 100% hands-on. All right, so just sit back while we dive into the session. Okay, so while I'm sharing my slide, presenting my slide so that we can have a review of what we did yesterday. So I will love that you guys have a questions. I don't know if you have a question concerning yesterday's session. So I would like you to drop the question very quickly at the chat room on YouTube. So just drop your question. Probably you have a question, you have a challenge, you have anything, especially concerning Genius software that some of you were able to get. All right, so just quickly drop your question while we dive into the practical session. So I just want to retouch on what we did yesterday based on your question. So I think someone is saying that all plugins install except PDB structure aligner. Okay, that will be on Genius software. Yes, don't worry about the PDB structure aligner. You really necessarily don't need that actually. Okay, so just don't worry. Okay, so Batmos says that I've not been able to install the software. It is telling me invalid key. All right, so the reason why you have invalid key is because the license name you are using has .txt. Make sure you remove .txt from the low GDP discount, which is the license name of your data. Do you understand? Make sure you remove .txt from it. So Blessing says about 5+ could not be installed. You can try it again. It could be your internet. Just make sure you go back and have a stable internet and try. One of the software that should not be installed should be PDP structure aligner, which you really don't need that actually. Okay, so there are other plugins that will do this the similar work that PDP would do. So you just ignore that. Okay, Helene said I've not been able to install the software. Is there a reason, Helene? Helene, is there a reason why? Helene, I would love to hear from you. Is there a reason why? Okay, someone says change your browser. Okay, we have another question. How transcriptomics and replication are called gene expression? Oh, transcriptomics and replication, why they are called gene expression. Now, the reason why transcriptomics has a very key point when it comes to gene expression analysis, I'm talking to a main race, all right? So the reason why there's gene expression in transcriptomics is because of in transcriptomics, we actually deal with RNA, and there's something called splicing in RNA. So splicing is the removal of introns. Introns, there's something called exons and there's something called introns. Exons are regions of nucleotide sequence that code for a functional protein. Are you getting it? So exons are regions that actually code for genes. All right. You know, we know that DNA is being transcribed to RNA, and that is the change of thymine to uracil, right? But you see, when thymine changes to uracil, we have normal RNA. But when it changes to mRNA, splicing takes place, and splicing is the removal of introns. Introns are regions that does not code for a functional protein. Exons are regions in nucleotide sequence that code for a functional protein. You understand my point? That code for genes of functional protein per se. So what I mean is that exons are those that are translated to protein. So when we say there are certain nucleotide sequences in a gene, are you getting the point? That code for a protein, it is not all the nucleotide sequence that actually code for a protein. The regions in the genes that code for the function of that particular gene, which is the protein, it is actually called exons. So the region that does not code for functional proteins are called introns. So when DNA is being transcribed to RNA, you have, you still have all the nucleotide sequence. But when it changes to mRNA, the introns are being removed by the function called introns. Splicing. So splicing is the removal of what? Introns when RNA is being what? Is being changed to mRNA. Are you getting it? It's being converted to mRNA. Are you getting it? So the mRNA are basically exons because those are what code for a functional protein. Is that very clear? Okay, someone says I broke up yesterday. So somebody was saying that yesterday session was interrupted. So don't worry, we will continue from where we stopped yesterday. We will continue from where we stopped yesterday. I hope the gene is okay. So the reason why we now have the mRNA is it actually has something to do with gene expression. You understand my point? So when splicing takes place, introns is being removed. We now have exons, which has become mRNA. So it is in that particular regions that genes actually come to expression. So, and that is the study of transcriptomics. That's transcriptomics for you, which is a study of RNA, everything that takes place in RNA. So in gene expression, we get to know how genes are being expressed, how certain genes are being knocked down, overexpressed or underexpressed or knocked down. Are you getting the point? Because that is the entire research when it comes to transcriptomics. Do you understand my point? And in the aspect of gene expression analysis. So basically, we want to understand genes that are expressing or that are not expressing, that are knocked down or that are underexpressed. Are you getting that, or overexpressed? So that is it basically. So now the reason why we study gene expression in transcriptome is basically because that is the region where we have the mRNA, the exons, and which actually code for a functional protein, because that's their region of expression. Actually, that's the point of expression. Okay, so what other questions do we have? So like I said, we broke up yesterday. It was because of some technical issue we have at Google Meet. So we are going to continue from where we stopped yesterday. Robertine, you said it says license has already been used. You shouldn't say that. Can you probably go offline? Okay, make sure you are disconnected from internet and try to install the software again. All right, Robertine. Robertine, I'm telling you, there could be some glitches. Okay, so at that point, so make sure you are offline and then you install it while you are offline. Is that clear, Robertine? Fogan? No. Deborah, an email will be sent on certificates at the end of today. So basically, between now and Monday, you will all get your certificate because we need to make those certificates. All right. So latest next week Tuesday or Monday, let's say Monday, all certificates will be sent to you. All right. For those of you who paid for certificates on Monday. So we're going to make the certificate this weekend. Okay, so what other questions do we have? Yeah, I think you can, for Mac users, probably you can, a link will be sent to all of you. So you should be able to see the software for Mac to download for the Mac application. Okay. Plugins are, if plugins are not showing, you need to be connected online for plugins to show. All right. You need to connect your laptop on the internet, then the plugins will show. When installing the plugin, you must be online. Okay, so what other questions? No. I've not shared my screen yet. So I will share my screen shortly so that we can dive into what we stopped at yesterday, okay? Okay, what is the meaning of NCBI? Somebody will ask you what is the meaning of NCBI. It means National Center for Biotechnology Information. Okay, someone is asking how do you make payments? Please chat the admin up on WhatsApp. Queen Raymond, I'm talking to you. Chat the admin up on WhatsApp. Okay, so let's dive into the session we have for today. So I'm going to show you around how you can actually I'm coming, give me a minute. How you can actually download the software. So we're going to start with downloading of the software tools, then we dive into the practical session that we have for today. We have a whole lot to cover. We have a whole lot to cover today. I think we still have more questions. Please, probably you should try to off your laptop and on it again. Maybe that is why you cannot connect to the internet. All right. So switch off your laptop and on it again and try it again. If not, just chat up with one of the admin. Okay. Chat one of the admin up. Okay. You said my computer wasn't compatible. Couldn't find one compatible with 32 bits. Oh, you have 32 bits. Okay. I will see into that. Can you chat the person you get you paid to, one of our staff that you paid to? I will ask him to send a link on how to download 32-bit to those who could not download the software because it's 32-bit. Just chat the person who you chatted with before you made payment to. Okay, one of our staff that you made payment to. Is that clear? Tawanga Venji, right? I hope I get that name right. Glory, what's your question? Okay, if I import a file, my system asks if it should be in CSV Excel sheet, or make sure the file you are importing is is a nucleotide sequence. Okay, so if you are installing a nucleotide sequence, it should not tell you, it should not ask you these questions. You should just import directly. Okay, I'm talking about Genome software. No, you cannot download the software on your phone, please. It has to be on your laptop. No, I'm not doing anything yet. Okay, so I'm just going to share my screen right now. So just give me a minute while I do that. Yeah. Please, I'm still working on trying to share my screen. Kindly exercise a bit patient, okay? So do we have any question while I'm sharing my screen? Okay, yeah, you can pay before Monday for the certificate. You can pay after this session. You can all get the software after this session as well, okay? So just wait for this session, okay? So the link to what, please? Arabic Gabron. What are you talking about? The link to what? Is it the link to the WhatsApp group or the link to what exactly? Please, I'm finding a difficulty in sharing my screen. Kindly exercise more patience, okay, so that we continue from where we stopped yesterday. I'm still struggling to install. To install what? To install what exactly? Hello, I'm with you. Marta, what are you struggling to install? Is it the Genome software, right? Is it the Genome software or the plugins? Yeah, we'll send the presentation to the WhatsApp group or to your email as well. Okay, yes. Yeah, we are going to send certificates, okay? But certificate comes with what's it called right now? An administrative fee of $3 only, and then you get your software, the activation key, the lifetime activation key of Genome software as well. Please, I want to be sure if you can see my screen. Please, can you see my screen? Can you all see my screen? Faith Samuel, what's your question? Please, can someone answer my question? What's your question, Faith Samuel? No. The recorded video, Faith Samuel, it's this YouTube channel, okay? So you definitely see the video on the YouTube channel. Is that clear, Faith? Okay, so let's look at the overview of what we done yesterday, and then we continue from where we stopped. But I'm going to be starting with with how you can install your Genome software. Okay, so for those of you who have issues with installing Genome software, we're going to start from there. All right. So this is the bootcamp agenda which we started yesterday. This was the theoretical session. You can go through the session 1 and session 2 that is still on the YouTube channel. Okay, it's still on YouTube channel. So you can actually access that. I mean, yesterday's sessions, okay? There were two sessions we had, but the second session was disrupted. So we continue from where we stopped yesterday here. And now it's on Junior Software. But let me start by going through the overview. So yesterday we learned in genomics and bioinformatics, basically looking at basic molecular biology, understanding molecular terminology, significance of genomics, the role of bioinformatics and career opportunities. We also cover introduction to DNA sequencing techniques, introduction to retrieving and handling NGS data from public database. We also look into introduction to bioinformatics tools for data analysis. I think we, under the bioinformatics tools for data analysis, we're talking about Junior Software, and I was taking you through data importation, creating of folders, and also we did some little DNA sequence analysis, all right? But definitely we're going to work around that to see more of basic DNA analysis on Junior Software before we begin to dive into other advanced aspects of the session. So in the practical session, we've been able to do hands-on retrieving and handling NGS data from NCBI public databases. You can actually learn more about that in yesterday's session on YouTube. And then we have hands-on Junior Software, which we have done. So today we're going to be looking into more of the basic sequence analysis, which is from where we stopped yesterday. And then we're going to look into designing effective primers for PCR and sequencing project. We're going to look at Junior Software and other one other tool that we can use to design PCR for the purpose of, we can design primer design for the purpose of PCR and sequencing projects. Okay, so you learn what primers are, what are primers, what was it called, all right, and why do we need primer when it comes to molecular biology. So we also look at performing in silico cloning experiment. We're also going to look at identifying and analyzing motif and repeats. We're going to see how we can get motif sequences and repeat sequences from within DNA sequences. So we also look into predict open reading frames in genomic sequences and their functions. We also look at utilizing species finder and P2RP for species identification and regulatory protein analysis. So we're going to be doing quite a lot today. So, but first, let's start with how you can install your Junior Software. So how do you install Junior Software? That is the first thing. How do you install Junior Software? So for those of you who are still having issues with getting access to Junior Software, just go to Google and search for Junior Software previous version, okay? Previous version. Did you see what I said? Junior Software previous version, okay? Or you say the previous version of what? Of Junior Software. So just click the first one, download Genius Primer, whatever, but don't worry. The previous versions are there. Okay, so you can see download Genius Prime 2024. So this is the latest version. So, but we need to look for that specific version we gave you because that was the activation key we gave you, which does not have much different with the latest version. The latest version is actually built in such a way that you won't actually get maybe lifetime action for it, but there's only little difference between the latest version and the previous version. They actually does more than 90% of what the latest version can do, all right? Because the only difference is just the plugin. Since the plugin on the latest version can also be installed on the previous version, it means it can actually does the same analysis. Is that clear? So don't have a doubt. So just to clear your doubts, okay? So to get access to the actual version we're looking for the installation of Genius Software, you click all products, or if you don't click all products, just scroll down. So you see version 2023, 2023.1, 2022. So these are the versions that are available. So you just keep scrolling down. This was 2022. So you can see next, right? You can see next right here. So you click next. What you can see here, you know, I'm showing you next here. So you just click this next. Yeah. So I've clicked mine. Let me zoom back. Yeah. So so this is 2022.1, 22 point, I mean 2020.0, right? 2020.1, right? So we have 2019. So we're looking for R9.1 or R9.0. In case you try R9.1 is not working for you, please try R9.0 or R8.1. It should work for you. Okay. In case if you try any other version that is not working between these, you can try this one or 8.0. It should work, and the same activation key should also work for you. All right, so I hope you get that explanation. So how do I download this? Let's go with R9.1, but don't forget, when you try other versions, lower version is not working, just try more lower versions, it should work. And still with the same activation key you get, that should also work. So this is Genius R9.1. So I've just clicked it, and it brought me to Genius R9.1. So I can click download for Windows, download for Mac OS, or download for Linux. For those of you using each of these any of these what's it called right now, OS, all right? So you can use for Windows, Mac, or Linux. Okay, so now let's click download for Windows. So I will click it, and it's going to start downloading. Can you see? Is downloading. So probably what this is actually downloading is 62 bits. So if you have any challenge, we can actually send you the link to download the 32 bits. All right, so just chat with the admin that you made payment to for the certificates, and then he's going to make that provided for you. All right, so by the time you download it, all right, so I don't know if I have Genius software here. I believe I should have a Genius software here. So once you download it, you need to install it. Okay, I don't think I have a Genius software here. Okay, so it's just that the downloading could take some time. So, but let me just resume it. So it's downloading. So I'm downloading the Genius software right now. All right. So to get the activation key, like we said, you have to pay $3 for the certificate, because the package for software tools come together with the certificates. All right. So, and the software is worth $5,000 for the lifetime access, but we have decided to give you all for free as far as those who get the certificates. Okay, it's just $3. That's just it. So, but the software is attached with the certificate. That's just it. So just kindly have it in mind that we are doing this on the basis of impact. All right. That is why we're doing this. So if you need the 32 bits, please, we're going to send the whole information on the what's it called, on the WhatsApp group. So just chat the person that will send it, and then the 32 bit will be provided. Okay, one of our staff. Yes. So another thing is that, please, the key that we'll be sending to you, it's specific for your system. Probably when you lost your system and you need to get the key, you could like maybe reach out, but and that would be like in years time. So just try to guide it jealously. I think somebody was saying she doesn't have access to Genius. I mean, she's telling her it's restricted. If you share your activation key with someone else, yours could be restricted. That's just the challenge. So just make sure you keep it to yourself. If it tells you and we have to meet with you online and try to activate it, and I still saying the same techniques, you might have shared it with someone else, and that would have made you lost access to it on your laptop. Okay, so it's set per user. All right, so I will ask the person in charge of the software, since some of you want to get access to the activation key. Please, where is Victor? Tell him to send the details on how to get the activation key to the WhatsApp groups. that some people wants to get the activation key. All right, so let's continue. So by the time I think it's completely downloaded now, so I want to install it. Okay, I'm going to go ahead to install the Genius software. All right, so let me get my download. So this is the software. So I just need to double click on the software and it's going to start installing. I just need to click yes, and it's going to start installing. So that's just what you need to do. So you need to install the software. I need to cancel this because I already have Genius software. So when you are through with installation of Genius software, it could ask you to activate the Genius software or click trial. All right, or click trial. Maybe you want to use trial. So probably the trial may not work. So that's why you have to get the activation key. So you could actually install the activation key right there, or you cancel the activation and come back here to search for Genius software like this. Okay, I'm searching for Genius software right now on my laptop. So it's searching it. So after you install it, you can cancel the request for you to activate the Genius software, or you do it that way. But I'm going to show you what you need to do. So even if it tells you right away to install it, you understand my point? I'm still going to show you how to install it. Anyways, works. Okay. So I'm going to click it. I've searched for Genius software for my laptop. I'm going to click it. I can actually bring it to the clipboard if I want. So now I've clicked Genius software. I'm launching the Genius software right now. So when you launch the Genius software, it will ask you again to install it. Okay, but I've already, I'm running my Genius software right now, and I'm waiting for it to open so that, so right now, while you're opening Genius software, it could ask you to activate it. So, but you could cancel the activation so that you can activate it later after you have opened the software, or activate it right away, whichever way works. But I'm going to show you how to activate it right now. Please, Moyo, someone wants to join the WhatsApp group. Can you send the 10.0 WhatsApp group? Moyo, can you send the 10.0 WhatsApp link to the chat box on YouTube, so that those who are not on WhatsApp can join the WhatsApp group? Okay. Please, can you tell Moyo that? I don't think she hears me. All right. So this is the Genius software. I've opened the Genius software. Now I'm going to show you how to activate the Genius software, because we have a lot to cover today, right? So how do you activate Genius software? Either when you open it and it tells you activate, or when you cancel it or you open it and you see restriction, you could see restriction here, and you just need to activate it. So how do you do that? So just click help, go to help. Can you see help here? I click help, and when you scroll down, you see activate license. Can you see activate license? So you go to help, or after you have opened Genius software, right? You go to help, you come to activate license. So this is the license. All right, this is how you activate the license. Now the name of the file you are going to be getting is the license name, and the name of the license name is just low GDP discount. Low GDP discount. That is just the license name. Some of you could see .txt. Make sure you remove .txt and use low GDP discount only. Do you understand? Yeah. PDP structure aligner on the plugins may not work, please, Mohammed. So just kindly ignore, all right? You don't need that. So just ignore it. You don't necessarily need it. Is that clear? Don't forget, the license name is low GDP discount. Everything is small letter. Make sure you remove .txt. That way it will work. Now, the license key is now everything in the file that we sent to you. When we send a file, the file we send to you is specific to you, right? And everything in that file is for you to activate the license. So I already have my own license here. I'm going to look for it. So this was my own license for the software I have right now on the system. So I'm going to open it. Now I'm going to copy everything in this file, everything in this file. I will highlight it, Control A, and then click Control C to copy it, and then go back to Junior Software and place it at the license key box. Space, rather. Can you see the space for license key? I am pasting it right now, and here it is. Can you see that? Here it is. All right. And then I'll click OK. And it's going to tell me it is registered successfully. I repeat, it's going to tell me that it's registered successfully. Definitely to tell you as well that it will be that is registered successfully. All right. So mine is done. So you can actually come to help, click activate license, and do all those things. Use low GDP discount, everything in the folder. You click it, open it, highlight it, and paste it here. Everything in the folder we send to you, and you click OK, and it will be activated. Or when you open it, it tells you activate license, and you click activate license. It also brings this box for you, and then you do the same thing. Is that very clear? Is that very clear? Is that very, very clear? You can say yes. You can just reply with a yes if it's clear, because we're about to continue from where we stopped yesterday. All right, so yesterday, at the download, we from NCBI, we actually download 4 sequences. All right, so 2 whole genome sequences, 2 sequences, sequences from 2 different organisms, rather. Sequences from 2 different organisms. You can download sequences for plants, animal, human, whichever one, okay? I just decided to work with microbial. So it doesn't necessarily mean you cannot work with other organisms, all right? So I downloaded for E. coli, sequences for E. coli, and I also downloaded sequences for Staphylococcus aureus. Now, the sequences I download for each of these organisms, I have a whole genome. I also have, what's it called, for gene, right? For sequences for just a gene. Is that very clear? Is that very clear? So, now I went ahead to open Junior Software. I click local and then click file. So before you can, because I want to create a new folder, right? I just wanted to create a new folder. So I must always click local, go to file, then click new folder to be able to create a new folder. And the new folder I can create right now could be DNA sequence analysis research, or research on DNA sequence analysis. All right, research on what? DNA sequence analysis. And I click OK. I click OK. Can you see? Research, and this is the folder, right? Let me try to draw this out. Research on DNA sequence analysis. So this was the folder. The next thing I did was go back to file, still on this folder, go back to file, come to import, go to from file, click from file, because I want to import from file. So I now go to PC, go to download where my samples are. All right, go to download where my samples are, and I click look for the sequences I downloaded, right? So I need to search for it. So the sequences I downloaded was this, okay, sequence 2, 3, and so this were the samples I downloaded, the 4 samples. Can you see that? So when I'm downloading when I'm uploading all 4 sequences, it tells me to either create sequence list or keep sequence separate. So I'm deciding to keep sequence separate because they are all separate sequences. So I'm going to click keep sequence separate. Michael Ward, please. Okay, don't worry. It will be sent to you. Okay, the staff will send you the file to the activation key. Don't worry. You can get access to this video on YouTube. All right, so that you can go through over it or again. So both yesterday's session and today's session, you can actually get access to it. All right. So can you see? So I've imported the first 2 sequences. So it's still asking me to either create sequence link or keep sequence separate. So I'm still going to click keep sequence separate for the rest of the sequence. Now I've uploaded all 4 sequences that I was able to download from NCBI public database yesterday. And these are the sequences. These are the sequences. So I can decide to view all the sequence. This is all the sequence. So whenever I click anything here, I see those sequences right on this box. And these are the details of some of those sequences. Can you see? Right here, I see the details of some of those sequences. So I can decide to click the sequences one after the other. So let me start by clicking the first sequence, which is Staphylococcus aureus MECA gene. Yesterday, we were talking about MECA gene, which is the gene that codes for resistance genes, right? It's the gene that codes for resistance gene. Now, this particular file that I downloaded is a gene bank file. There are different file formats in bioinformatics. The FASTA file just contains the DNA sequence of the organism, while the GB file, which is the gene bank file, contains the annotated elements of the part or that made up the gene. You understand my point? Or that made up the sequence, the annotations that made up the sequence. What I mean by annotations are regions that code for a functional protein or regions that code for a specific function or structure in an organism. You understand my point? By the gene that code for it. Is that clear? Someone was saying, Chinedu was saying it was not clear on downloading of Genomic software. Please, you can go through the recorded session. You can go through the recorded session to know how to download the Genomic software again after this session, okay? You'll find the video on YouTube. Thank you. So what am I saying is I have 2 kind of file for Staphylococcus aureus here, all right? I have the FASTA file for this particular gene. Can you see it's the same gene? This gene, you can see it's just the sequences, all right? It just contains the sequence. Let me remove this translation. When I click this translation, I see this translation sequence, right? So I'm removing this translation, I'm removing this wrap. So the reason why I remove this wrap icon is because I want to see, can you see, I want to view the sequences vividly. This was what we did yesterday. I want to see the sequences vividly, right? So I can zoom by clicking here. I can zoom out. I can zoom in so that I can see the sequences. So these are the sequence that code for this particular gene that is responsible for antibiotic resistance in Staphylococcus aureus. But I am viewing the FASTA file of this gene. The FASTA file contains only the nucleotide sequences of this gene, not the annotated features. Is that clear? Some of you are talking about you have not received the Genomic software. Have you made payment? If you have made payment, please do not worry. Just make sure you chat the person that you made payment to, one of our staff you made payment to. So the staff is going to send you the details. So just please don't worry. At the end of this program, between now and tomorrow, we're going to make sure all those things gets to you. All right? And you can always reach out to us on WhatsApp. All right? We're happy to help you anytime. So just make sure you are focused on the session, even though you go through the recorded session later on. So thank you. Now I can view the translation region. So by the time I click translation, it's translating the entire sequence, this entire sequence to give me regions of proteins, of amino acid, right? That each codons code for. So you can see TTC code for F, which is phenylalanine. This is a codon. This is another codon. So ATA code for what? Isoleucine. So that's how I see the translation of this sequence. I can see the complementary strand. You know, DNA is made up of double helix, right? So I can see the complementary strand of this sequence by clicking complement here. When I click complement here, it gives me the complementary strand. Do you see that? It gives me the complementary strands. So then I can remove it. Translation, I just click it, it gives me, I just need to count 3 codons, 1, 2, 3. 3 bases rather, 1, 2, 3, and it code for just 1 codon, which is F. And that particular codon, it code for encodes an amino acid. You understand my point? The codon encodes an amino acid, which is what? F. And that F is phenylalanine amino acid, which is a protein. Is that clear? So we have what? ATA is another codon after the first codon. After the second codon, we have another codon, TCT. Can you see? You can see the colors separate it. Can you see? The color separates it. The color of the amino acid separates or categorize each what? Each codons that code for them. The codons, which is what? The nucleotide bases that code for them. Thank you. So let's proceed. All right. So that's it basically. Now, I was talking about the annotated features. It's still the same sequence, but this time around, we're seeing the annotation. Can you see that? So what you see here is the annotation, and you can see the details of the annotation by clicking here. This yellow icon here. So you see that there's CDS1 and gene1. The CDS is coding sequence, while the gene, the CDS means coding sequence. The coding sequence is region of the sequence that code for a gene. And this is the gene. So whenever you see a coding sequence, you always see the gene. Do you understand? Okay, so let's continue. Now let's look at the whole genome. These are gene sequences. Now let's look at the whole genome sequences. So I downloaded Escherichia coli whole genome sequence. All right. And this particular whole genome sequence contains the sequences. This is a FASTA file of a whole genome sequence. And you can see how long it is, right? So yesterday I was talking about the sequence length. The sequence length of this gene is 156. And the sequence length of this complete whole genome sequence of Escherichia coli is 5.1 million plus. To get certificates. Okay, so we're trying to make sure that you guys have access to some of these things, okay? So now, this is 5.1 million plus, right, of sequence length, and the DC content is 50.6. So now, let's look at the annotated. So this is the whole genome sequence, and it has been annotated. So this is the annotated view, all right? This is the annotated view in circular form. We can remove the circular form by going to sequence and remove the circular form. Can you see? I've removed the circular form. Now I'm seeing the linear form. I can go back to sequence and click circular sequence, and it gives me the circular form of this sequence. I can maximize this space so that I can see it very well by clicking here, this icon here. Can you see? So I can zoom in to see the annotations. Can you see? I can see the annotation. Now, the reason why we are seeing many annotations here is because this is whole genome sequence. Whole genome sequence is the entire organism set of the organism. I mean, the entire DNA sequence of the organism. So in the entire DNA sequence of the organism, we have many genes. Is that clear? So now let's go back. So I want to remove the circular form so that I can have a linear form. And I can zoom in to see the annotations. Can you see the annotations? Each CDS code for a particular gene. Can you see? We have CDS. The CDS is in yellow color, and then we have the genes. So this particular CDS code for this particular gene. Some of these CDS are already named. Can you see? Some of them, this is the name, and this is their notes. It's an automated computational analysis. Okay. Okay. No. So this is the name of the CDS. So you can actually search the name of this gene online to know what that gene code for. But this is the product. This gene code for this product. Can you see? The HTH type transcriptional regulator. So this particular CDS that code for this gene is responsible for what? for this product, and the product is STH type transcriptional regulator. So that means this particular gene is responsible or associated to a regulatory processes or biological processes or biological function in Escherichia coli. Are you getting it? Are you getting it? So that is it basically. So we can look at another one. So let's look at another gene. Let's look at the first gene. This is the CDS, and this is the gene. So let's look at what this CDS code for. It codes for this gene, which is this gene. But what's the function of this CDS? The function of the CDS, you just go to product, and the product or the function is actually a chromosomal replicable initiator protein. You understand? The function of this gene is found in the CDS. So you just need to click the CDS this way, put your cursor on the CDS, and then this will show this is the gene, this is the local start, this is old local start, this is the go function. So GO function means gene ontology function, and it is actually responsible for ATP binding. It is associated to the ATP binding biological function, or DNA binding biological function, or replicatory region binding biological function in the organism. Are you getting it? That is what is associated with too. Are you with me? Are you with me? So that's it basically, right? And this is the product. It is associated to chromosomal replicable initiator. So it means it is responsible for initiating chromosomal replication in the organism. Are you with me? So that is how you get to know the function of a gene in the entire organism set, DNA sequence set of an organism, right? In the entire DNA sequence of an organism, this is how you know the product of each gene, the function of each gene in an organism. So even if you get a gene bank file of a human biological data, you can import it into Genome software, look around it, and be able to know the functions of each of those genes and what they are coding for. Annotations are regions that code for a functional protein. I repeat, annotations are regions that code for a functional protein. I repeat, annotations are regions that code for a functional protein. That is annotation. So when you get a whole genome sequence, you can annotate a particular region to know the function in that region. And we're going to be doing some annotation hands-on experiment in the course of this program. That is what annotation is. Predicting regions that are called for a functional role in an organism, DNA sequence. Yes, that's what annotations are. Somebody is asking what CGP island. What is CGP island? CGP island is just like a graph that shows the level of expression of a particular region based on probably the covalent bonding or the regions that cause for a specific functional feature, a functional protein. I repeat, CGP region is just showing you a graph of a region that is a potential region. You understand? In terms of responsible or coding for a functional protein. Are you getting? Let me repeat myself. In a whole sequence, we have regions that code for a functional protein. So when you get to a region, the CGP island increases because that region code is a potential region that codes for a functional protein. I hope you get it clearly. That's what CGP island is. Is that clear? So there's so many regions that code for so many genes and some other elements in a whole genome sequence. So when I click this file here, can you see? When I click this file here, can you see? Right? This icon here, it shows me everything that is present in the whole genome sequence of this particular bacteria, which is Escherichia coli. So we see the CDS. So there are like 4,848 coding sequences. So there are regions that does not code for anything. That region are called introns. So when you zoom, you can actually see a region that does not code for anything. So you just need to look for it. So there are regions that does not code for anything. I'm trying to look for it. Okay, let me reduce this. So I'm going to highlight a region that does not code for any functional protein. So can you see this region? There's no functional protein in this region. Likewise, this region, there's no functional protein here. So these regions are usually introns. So the region, so when you download a whole genome sequence that is annotated, a GBank file and uploaded, you actually see a region that does not code for a functional protein. Those regions are usually called introns. Is that clear? So in this whole genome sequence, we have what? We have regulatory sequence, we have repeat region, RNA source, we have miscellaneous, we have mcRNA regions. So we have so many regions here, and these are the numbers of those regions and their color. So you could just look for them around in the course of this while you are moving around. So you could just look for those things with their color, okay, depending on their color. All right, and that's a way you can actually get to know it. So you can click this icon and then click pop out, and then try to click them one after the other. Can you see? So you can click one after the other. This regulatory sequence. So I can click this, and then it shows me the regulatory sequence. Can you see? So this is the regulatory sequence. I can click it this way. I want to know the repeat. Come here. This is the repeat region. All right, because it's so much, we can't be looking for them in the entire table. They are too much in this table. So we have to click type, unclick them, and then begin to look for them. This miscellaneous, what was it all about? This is the miscellaneous, and this is the notes. You could actually get to learn more about what this miscellaneous is using the motif region. I think it's a motif actually. So we have RNA, 22R RNA. So these are RNA in an organism genome, and you can use this rRNA for phylogenetic analysis. You can get rRNA across all genome sequences and then use them for evolutionary study. Do you understand? We can get tmRNA. So this is tmRNA. Can you see that? We can get tRNA. There are 89 tRNAs. So just click any of them, and then you'll be able to get those tRNAs. So we have, I think we've checked repeat region. ncRNA is 5. So this is ncRNA. So this is how you see them. So I'm going to minimize this, right? Yeah. So clicking this. So I'm going to pop in. I want to remove it so that I can have all of this. Is that very clear? No, you are paying for the certificate, please. See, the program is free for everyone. I repeat, this bootcamp is fully funded, it's free for everyone. You are only paying for certificates, okay? It's free. Somebody is asking if he wants to pay for this program. It's free, please, except if you want to donate. We are open for donation. All right, we are open for donation, but it's fully funded, okay? Thank you. All right, so that is it for basic DNA analysis. Now let's dive into the session. So diving into this session now, let me remove this. So we are looking at the next thing is primer design. So I'm going to be talking about primer design. That's the next analysis. So in the next 10 minutes or 5 minutes or so, I'm going to be talking about primer design, and I'm going to be showing you how you can design primers. Okay, so what is primers? Okay, primers are regions and specific regions in the DNA sequence that you can actually use for the purpose of PCR. Please, Ayobami. Okay, someone will reach out to you. Have you? Someone will reach out to you, please, on the payment. Just calm down. We're going to reach out to all of you on WhatsApp for the payment. Please reach out to them. Thank you. All right, so thank you. We have a whole lot to cover. Please, I'm not going to be taking some of your questions for now, but I will get back to it later. Thank you. So we want to design primers. I repeat, we want to design primers. So how do you design primer for PCR? I told you, when you want to sequence a DNA from an organism, you need primers. All right. Now these primers can be targeted, or it may not be targeted, can be whole genome. Do you understand my point? So when it comes to primer design, when it comes to sequences, especially 16S or Sanger sequencing, you need primers for them. And these primers are usually classified as model template for sequencing, because you need primers for replication. Do you understand my point? The essence of primer is to replicate a specific region, to amplify a specific region in an organism. So now let's look at the FASTA sequence of Staphylococcus aureus gene. Imagine you want to your research is that you want to focus on this particular gene, which is MECA gene. In your research, are you with me? In your research, you want to focus on MECA gene. And this MECA gene is your research focus. Now you want to get this gene from maybe multiple cultures of organisms or plants or animal, whatever. You understand? Depending on the kind of gene you are talking about, okay, but this is bacteria for Staphylococcus aureus. So if you want to get genes from plants, you also need PCR and primers. So you just need to be able to do some tissue culturing in the lab and be able to extract the DNA. After extraction of the DNA, the next thing that needs to be done is design primer. Designing primer is usually in silico, is dry lab. Now, this is how you design primer for a specific gene. A lot of researchers are in need of primers. Some people pay to know how to design primer. Some people pay to people who knows how to design primer for their research. But here you know how to design primer and which good primer to pick. Are you with me? So this is a FASTA sequence data of MECA gene from Staphylococcus aureus. I'm a researcher. I want to get primer for this gene because I'm interested in this gene for my research, and I want to sequence it from the bacteria. By the time I get the DNA sequence, I'm sending it. I'm supposed to send the primer along. So now I've gone to NCBI, gotten a template or a model of this resistant gene. Now I need to design primer so that I can send it together with my DNA so that I can get that specific region also from my own cultured bacteria. All right. So now I have clicked it. You have to click the sequence you want to work with. All right. You know, right now we're not clicking anything, so there's nothing here. So now I need to go and click this, or better still, let me create a folder for it. So I'm going to click local, click file, and click new folder. I'm creating a new folder for this primer design. I call it primer design. All right. Primer design. I've clicked primer design. Now, where's the folder? The folder will be highlighted. Here is the folder. Can you see? Primer design. There's nothing in this folder, but I'm interested in a particular gene of Staphylococcus aureus, which is MECA gene. I've downloaded it already from NCBI. Now I need to go to file, come to import from file to upload that particular gene. All right. So I'm going to look for this gene. So I've downloaded it. So it should be sequence 2. It should be in sequence. Can you see? Sequence bracket 2.FASTA. The second one is GB file, but I don't need GB file. I need the FASTA file. I can also need GB file, so let me also employ the GB file because I need it to explain something to you for the GB file. So let me import it. So I've imported the 2 sequences, but I need to start with FASTA file. So the second one is the FASTA file that does not have any annotated features. I can zoom in to see the sequences. Zoom out because I've seen the sequences. So now I want to design the primer. So now let's go to design the primer. Do you see this icon here? Primers. This icon called primers. I click it. I've clicked that icon called primers, and I will click design new primers. I've just clicked design new primers. I repeat, I click the sequence that I've just imported. I come to primers, I click it, and I go to design new primers. Can you see? So now I've clicked design new primers, I'm here. So in primer, we usually need them in forward and reverse. I repeat, we usually need primers in forward and reverse, just like forward and reverse complement. You understand my point? Of a particular DNA sequence. You know, when we have the sequences, you know, the forward strand and the reverse strand. So when you are designing a primer, you usually have a forward and a reverse primer. All right. So by the time you see the design new primer is highlighted, then we have forward primer and reverse primer. Forward primer, reverse primer. So you can see task, right? The task. So the task is I need generic. I need it to be generic because I want to get primer across all the sequences. All right? Across all the sequences. So I just need this. All right? Now, include region. You can decide to reduce the region. If you are trying to design primer within a whole genome sequence, and you've identified a specific region in that whole genome sequence for primer, you can use this include region to minimize the region of the sequence you want to design primers on. Are you with me? Are you with me? Yes? I need a reply at the chat room. So we have what? Include region. So this is when you are working with whole genome sequence and there's a specific region you want to target in the whole genome sequence. That's it basically. So, but you can decide to remove it and focus on all the genome. Remember, we are using a very short sequence length of what? NECA gene. Product size should be between, I'm also picking from, because the whole size is 156, so I'm picking 1 to 156. I can decide to remove it as well and leave it at default or click it as well. So optimal product size, you can decide to, to increase the product size of your PCR, of your primer, but let's leave everything at default. Now we click OK. I have clicked OK and I have successfully designed a new primer. Right now, I have designed 1, 2, 3, 4, 5 primers. Now, let me show you something. Let me go back to primer again. Design new primer. You see, when I wanted to design primer, the number of pairs to generate was 5. And that is why I'm having 5 here. I repeat, when I go to primer, by the time I wanted to design the primers, I see the numbers of pairs to be generated should be 5. I can decide to say 1. I can decide to say 10. Are you getting it? So that is just it, basically. So that's just it, basically. All right? All right. So that is it, basically, right? So now we have 14F forward. That is, 14F. 147R. All right, we have 14F, 150R. Those are primers. 13F, right? 13F, 147R. 14F. So each of this F, this is what? Is primer. How do you know the primer? It will always be highlighted. Can you see if you trace it down, it highlights this. From here, it highlights its primer. It's reverse primer. So I need to extract this forward and reverse primer. How do I do that? So let me try to say something before I proceed. How do you pick the best primer? You must pick the best primer based on the region of coverage. Pick the best primer based on the region of coverage. Ideally, all these primers are very okay. Some of the primers, the regions are shorter. But the reason why we see the same primer on the same region is probably because of of the length of the sequence length. So ideally, we have a very longer sequence length. You have series of primers in different regions. You understand my point? But try to pick primer that covers quite a number of different regions within the sequence. Do you understand? Primers is used for sequencing, for extracting DNA sequence from a cultured bacteria or a cultured tissue of any organism. Are you with me? That is what we use primers for. So if you want to get DNA sequences of a particular cultured organism, you understand my point? You need primers for them. So it means you have to go online to get those primers. You understand my point? I mean, to get a template, reference data, and design primer from them and be able to send to the lab for sequencing. So the next thing we not just need to do is what? I right click. I need to right click on the first one. I can click anyone here. Anyone here is good because it covers the same region. So you got to click the first one, right click on it. We want to extract the primer. So I click extract regions. Can you see? So I've extracted the region and then click OK. So I click save. And now I have my forward primer. So I need to go back also to extract the reverse. Extract region and click it. Now I have my forward and reverse primer. So how do you save this primer so that you can send it to the sequencing lab? You just need to click it this way. Click the sample, click file, go to export, click batch export, and come to file format. I repeat, how do you save it? You click it, come to file, go to export, click batch export. You see file format, click file format. We are looking for faster sequence format. Can you see faster? I've just clicked faster. And now I need to see where it's been downloaded. I can choose where it should be downloaded. So I can decide to download it to desktop. Click OK. So I'm downloading it to desktop and I click OK. So it asks me, do I want the sequences to be in character, in whatever I want? So I just leave it at default and I click OK. So now I've saved this file. I can now go to desktop and search for my faster file that I just downloaded. This is it. This is I can open it and this is the faster file. You understand? Of the forward reverse. I need to go back now and what? Download the reverse. Click file. Go to export. Click batch export. All right. I need to pick faster sequence / alignment format. Okay. So I'm still exporting to dashboard. So it's there. I click OK and then click OK again. So now I should have my reverse. Can you see? This is the forward and this is the reverse. So somebody's asking, where is the primer required? Where is the primer required for PCR? Like I said, it is after you have done culturing in the lab and you need to get DNA sequence for molecular characterization for a research in school. So that is when you need to go and get the template of the kind of gene you are interested in the data. Mostly it is 16S data that you can actually get for those of you doing 16S analysis. All right, 16S RNA sequence analysis through Sanger sequencing. So you need to get the 16S RNA, bring it to Junior software, design primers, and send those primers to the sequencing lab in order to give you the DNA sequence of the organism that you are trying to do research on. Is that clear now? Thank you so much. So now we have done primer. There's another way to design primer very quickly. So let me take you to So there's another way to design primers, and it is through this link. All right. Let me say Amuser 1.0. So let me try to search it online. Amuser for primer design. I hope I see it. Okay, so this is Amuser can design primers that allow amplification of a sequence of vector backbone only once with one set of primers, and design the primers for the genes such that every single fragment will fuse directionally to the amplified vector backbone. So basically, Amazon can be used to design primers for amplification of sequence. And this is the link. So you just click it, and it brings up right to the link. Okay, that's how you get it. So this one is just straightforward if you don't want to use Genius software. Okay, so after you get that, you need to import your file. You need to choose your file. You know, we already download the file. Let me show you the file. So this is my own file that I imported yesterday, right? The FASTA file that I imported yesterday for this is it, right? For Staphylococcus aureus for this particular gene. So I can now come to, okay, I can now come to the online platform, online tool. Now this is online tool, all right, for designing primers, and I look for it. So I think it's this one, FASTA file. I click open, and I've already, so I can actually paste it. I can open the data like this, and highlight everything, right, and paste it here. Or better still, I just upload it. So right now I'm uploading. So I've uploaded. So I want linear, the output should be linear, all right? So PCR experimentation conditions. So you can decide the conditions of your primer. These primers, the condition is actually determined by the sequencing lab. They will tell you the conditions they are trying to use on their machine. These are parameters on their machine for the sequencing, for the PCR, for the that will be needed for the sequencing of that particular DNA sequence, right? So, but let's use default, because default is actually put at the best condition, okay, such that it could work at any time. All right. So then we submit our query. Don't forget we've uploaded the sequence, and I said the output should be linear. I leave it at default. That's step 3, and then I submit query. So now I've clicked submit query, and it's going to analyze my sample and give me my primer. So right now it's analyzing my sample, and it's going to give me my primer shortly. Is that clear? Is that clear? It's going to give me my primer shortly. Now the analysis is done. AMUSA query result. So this is my result. The number of fragments submitted was 1. I just submitted 1. And this way the condition, the salt concentration. Let me zoom in so that you can see it. The number of fragments is 1. The salt concentration condition was 50 nm, and the primer concentration selected was 0.05 All right, the linear cloning is not no cassette, right? It's just like some kind of backbones or so. Let me not dive into that in order not to confuse you. So the overview of the needed primers from 5' to 3' right? 5' to 3' rather. So we have the fragmented that was the fragment that was submitted. So the fragment that was submitted was this. So I think it actually predicted primer based on the forward and the reverse strand. You know, the primers that needs to be gotten needs to be in forward and reverse, right? So this is the fragmented sequence based on the primer sequence that we got. I repeat, when we submit a sample, we need the forward and reverse primer, right? So this is the strand, the forward and the reverse. This is the temperature condition that it was designed on, right, to give us the primer. Now, don't forget, primer should always be between 18 and I think 24. Primer should always be between 18 and 24 base pairs. So if you should count these primers right now, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. The other one is 18, so it works. So I think this should be the first primer. So this is overview of your final construct after cloning. So we have graphic overview of DNA sequence and primers. So this is the sequence, graphics overview sequence, like maybe visualization or something. All right. So in these sequences that was selected, this was the primer that was choosing. and this is the reverse primer that was chosen in the same longer sequence of the DNA sequence we submitted. All right. So this one is forward primer, and this one is a fusion region and reverse primer for joining whatever. So this one is for forward, and this one is for reverse, and that's how we got these other primers. But let's just proceed. These are the details of the primer. This is the forward primer, and this is the reverse primer, and these are the overview details. This is the final details we got. So the GC content, the GC clamp present, all these results or information you need it actually for your research project. Is that clear? You need it for your research project. And this was the sequence we submitted, right? Can you see this was the sequence we submitted, or what's happening? Oh, I think I submitted the, it's as though I submitted E. coli whole genome sequence instead. I think it was whole genome sequence I submitted instead of yeah, it was E. coli I submitted whole genome sequence to design primer for it. Yeah, it was not Staphylococcus aureus, but that's okay. Just to tell you that this particular platform is not limited to just shorter sequences alone. You can actually predict primer from whole genome sequence using the platform. Is that clear? Everyone, is it clear now? So that's how you get your primer, and these are your primers. So you just need to pick those primers and save them in text document, and then that's all. All right. So that's all with Amuser file online faster online tool to predict primers. All right. So that's it. Now the next thing we need to look into right now is what? Perform in silico cloning. So we're going to use Genome software to perform in silico cloning. Right now we're going to be very fast because we don't have much time remaining. So how do we perform in silico cloning? So what is cloning? Cloning is usually used maybe when you want to carry out some biotech research, and then you want to see the dry lab or in silico cloning, how it's being used, the regions it will be by the time you are doing the wet lab cloning. Do you understand my point? So the dry lab cloning is just so if you want to design primer for cDNA sequence, you just need to get the sequence and import it, okay? And then you design primer just based on the procedure I've provided. Is that clear, Satkan? Thank you. So for those of you in power technology, that's why we are showing you how you can design, how you can do cloning right now. So I want to show you how you can design cloning. So when you want to do cloning, you need plasmid, all right? You need plasmid. These plasmid are vectors, all right? These plasmid will be vectors. Vectors are something that houses what you actually want to impute it, right? So let's try to look for. Okay, so this is plasmid, and these are vectors. All right, these are vectors. So there are a lot of vectors. You can actually go on NCBI to download vectors, but there are some of these vectors we can actually get on on Genome software. So these are what? Everything I'm clicking here, you actually find it under sample documents. Go to sample documents. Sample documents. So when you go to sample document, you come to plasmid from what? From NEB. There are 27 plasmid here, but you can actually get plasmid from NCBI, just like the search will show you. So you can just go and search for a specific plasmid on NCBI. You download the gene bank, and then you'll be able to have this plasmid. You can import them, and then you have them as plasmid. But there are many plasmid here, and this is these are some of those plasmid, right? So, and these plasmid are classified as vectors. I repeat, they are classified as vectors because we need them to impute, all right, to impute regions where we want to clone, all right, in a specific organism. We infuse those regions into the vector. Are you getting my point? We infuse it into the vector. So now let's go. This is the first sequence. So let's try to create a new folder for cloning. New folder. Let's call it cloning analysis. We click OK, and this is the folder. So, but I need to import a vector. So let's come here. I'm looking for a vector that I can work with. Okay, let's work with this vector. Complete sequence. I've copied it. I'm going to paste it here. So I've pasted this vector. All right. Now you can decide to actually infuse primers into a vector. I repeat, you can infuse primers into a vector. I repeat, you can infuse primer into vectors. All right. So let's see. So this is the plasmid. Okay, let's get a primer. You can actually get any sequences. Probably, I think it would be better if we infuse a primer, right? You could actually infuse a specific region of interest, right, into a vector, all right, because it's used in the biotechnology. Some of these, what's it called right now? Some of these industrial work in the industry, like probably fermentation, or if not fermentation, I'm talking about there are certain enzymes that have been created, industrial enzymes that have been produced through cloning. The reason is because they have to get some of these regions in a specific plant or maybe in a specific organism, and they want to produce it to be multiplied. You understand? They want to multiply this specific region, this specific enzyme. They can actually infuse that particular enzyme through cloning into an organism for replication. You understand my point? So that is what cloning is all about. But this is just the in silico aspect of this work, and that's what I want to show you right now. So let's get the primer. Let's get a forward primer. So I'm copying the forward primer. I'm going to the cloning folder. I'm pasting it. So I just paste the cloning folder. Now this is the cloning, right? So we need to infuse. All right. All right, so this is it. All right, so there are different cloning procedure. We have topo cloning. Can you see that? We have golden gate. Some of it needs some kind of analysis, but we're just going to use one probably in the next set of our work. We're going to look, we're going to see how we can perform the analysis. Okay, so let's use restriction cloning. Restriction cloning restricted to a specific site. So this is vector, polylinker region to cut within. So this is just this particular genome software is designed for that. So we have insert forward into, all right, no restriction site or overhang, inserting entire sequence. So right now we've been able to infuse a specific we have been able to infuse the forward primer into this particular vector. Are you getting it? So how do we know that? So you just minimize and come to the cloning view, and then come to the annotation part. You will see inserted sequence. Can you see inserted sequence? And this is the color. So you look for the red color in the whole of annotation view of the vector, and then you can see it. Here is it. Can you see? Let me try to zoom. Let me try to zoom. Can you see it? Okay. Can you see it? So let me zoom more so that you can see it better. Yes. So out of all the annotated features of this cloning, we've been able to do a kind of restriction cloning by infusing, all right, that particular primer into the sequence. And here it is. It's been attached to the cloning at this particular region. Like I said, this is just in silico cloning to give you overview of how your biotechnological cloning in the lab would be like. You understand my point? And this is it. Most times, some of this cloning is actually being done, and then together with the vector and the sequence that has been infused can actually be synthesized in a synthesizing company for some other biotechnological work. So, but this is how you do in silico cloning. Is that clear? So today we'll only be able to do restriction cloning for now. All right. So probably in the next time we'll be doing much of the other cloning aspects of our work. Hello, everyone. I hope you are all following. Hello, I hope you're all following. No. The vector is like plasmid. Vectors are like plasmid, okay? They can actually be found. They can actually be found across, maybe by nature in organisms, okay? So probably bacteria and all of that, but some of them are classified as plasmids. Is that clear? Is that clear? So that's it basically. So the next set of things we have to do right now after cloning is identify and analyze motif and repeat region. So let's quickly go to NCBI, Genome software. So we go to research on DNA sequence, and we click one of the sequence. So we want to get motif, right? I repeat, we want to analyze motif and repeat region. Motif and repeat region are short regions that code for a specific element in an organism. Motif are short regions in a specific sequence that code for a specific element, okay, in that particular sequence. So right now, let's use the gene bank, or maybe let's use the FASTA file to look for repeat regions and motifs. So how do you do that? You go to annotate. Can you see annotate? And then you can search for motifs. So I can click search for motifs. All right. So I'm actually searching for motif. We say there's no motif found in this sequence. So let's do that again. Search of prosite motif. There's a specific motif in an organism you want to get, or specific annotation name you want to find motif on. But if you want to get it on an entire sequence, you just leave it that way. So it seems the sequence is too small for us to get motifs from. But meanwhile, let's use whole genome sequence of FASTA file, okay? And then let's search for motif from the whole genome sequence. Don't worry, guys. You can watch through the video session and then you'll be able to practice on your own. So let's go to annotate and predict. And then we come to search for motif. Now we are doing for whole genome sequence. So let's search. So we're searching for motif still. It seems we find no results still. Okay. It seems we could not find motifs by default, but there's a better way we can find motifs. Now let me give you how you can search for motifs. So motif could be a specific region in an organism that is of interest to you. So one other ways you can actually get motifs is, let's go back to the short sequence because of our time. So let's come to search for motif. Now, I can actually impute a specific region, all right, to I just need to include maybe how can I say it now, a sequence, a very short sequence length of nucleotide bases, all right, a very short nucleotide bases, a very short sequence of nucleotide bases. So it could be in 5, it could be in 10, and then we're going to impute it to search for that region in the sequence. So there's a default way. I think the default way is not working probably, I don't know. But there's the other way is you need to know the region, all right, to be able to get the motif. So motif are regions of interest in a specific region, all right? So let's say you want to find a specific region, maybe there's a specific region of interest that you're looking for. So that is actually called motif. And then let's say you want to get CTAAC. You understand my point? Maybe this is a region and you want to look for it. So you can search. Still We don't have it. So you need to actually know a specific motive you are looking for. Let's say TAT. Okay, we find TAT. Can you see that? So it means we found 10 TAT in this short sequence that we have. There are 10 of it. Can you see from this annotation view? These are motifs. Don't forget, I say motifs are regions that code for a specific region. I mean, that are regions of interest. Some of these motifs could be regions that are responsible for a mutation or variation associated to a particular disease in any organism. Are you with me? They could be associated to a particular disease in any organism, either plant, microbial, human, whatsoever. So maybe from other previous research you've gotten, you discover that, okay, there are specific regions, mutation in a particular region, in a specific gene, all right, that, is associated to a particular disease. So maybe what is actually associated to the disease is TAT. So in a normal organism genome, there's supposed not to be TAG in a specific region. But for you to search if TAG exists in that particular sequence is by using the motif search. Yeah? We can actually search for another one, maybe CAC. So let's say CAC. Remember, your motif should always be in the nucleotide basis. I think we found CAC. We found 2 CACs. So these 2 CACs, based on your previous research, could be like, oh, these CACs in this particular gene, whenever they occur, probably at a particular region, it means that they are associated to a particular variations that, resulted to a particular disease or cancer or whatsoever. Are you understanding? Are you understanding? That is what motifs are. That is the essence of motifs in research. So by the time you understand a specific region, that this region is associated to a disease, you get that particular region. You get the nucleotide basis in that particular region. It could be TAT, CAC. Then you can now get other genes of the same research. I mean, of the same gene you are looking for. Other genes from other people, other patients, of the same gene you are looking for. Then once you find those motifs in them, it means they existed. It means they share a common disease lineage or whatever, or association, a common disease relationship or association within those patients. Those are the essence of motifs. No, I already told you, the use of motif is to search for a specific region that is associated to a particular disease. Are you with me? The essence of motifs is to search for a specific region that is associated to a particular disease in an organism. Are you with me? So when you have a particular motif that is associated to a disease, you can search for that same motif in other sequences of the same organism. So if they existed, it means they have the same relatedness when it comes to the disease. You can search more on motif, but I've already shown you how you can do the analysis and get motifs, okay? So the next thing is, I'm going to unclick this and remove them, okay? Repeats. I want to show you how you can get repeat sequences in a particular organism. Sorry, I'm coming. I'm coming. I'm looking for something. Okay. So I think the next thing we should look into, because of our time, because I can't find the repeats here. I don't know. All right. Okay. Okay. So let's go to the next thing. It seems like I can't find repeats. I'm actually looking for a way I can get repeat sequences on okay, so let me show you the other way around. So the other way around to actually get some of these transnational gene element, probably we'll be able to get repeats from them, is we can actually use this option, right? So this show us the annotated features, but to get more annotated features, we can come here and click annotate from. Can you see that? So but we have 0 results, so we need to click advanced and reduce this length probably to 6, the index sequence length that we're looking for, to 6, and then click OK. It's still giving us 0, but we just reduce the similarity ratio, so we should be able to have something right now. Okay, so we have 38. I don't think we have repeat sequences in this particular data. Let me reduce it again. How you can get more future? The more you reduce it, the more you get the results. Okay, promoter, tag, regulatory, localization. So let's go back and then you click apply. So you should be able to see the genes within the same sequences. Localization, motif, we have more motif, right? That was the motif we designed the other time. The promoter region, the regulatory sequence, tags, and terminator. So okay, so that's it. That's how you get all that genetic features. So let's proceed. Because of our time, I don't want us to waste our time. So we have predict open reading frames. So you can predict open reading frame. Let me remove all the annotation. So come back here, click find open reading frames. Find open reading frames. So clicking find open reading frames shows zeros, but we can reduce the size to like maybe 10 and apply. And then click again. Can you see we use minimize size to be 10, and then we're able to find open reading frames. Open reading frames are regions that code for a functional protein. I repeat, open reading frames are regions that code for a functional protein. Yeah, the regions, I mean, the motif could code for an amino acid. I'm answering, the motifs could code for an amino acid, but they may not code for a gene, okay? So, but they could code for amino acid within a gene. But that region is actually associated to maybe variation, mutation within an organism that are associated to a disease or associated to anything you're actually looking for, maybe resistance or whatsoever. Do you understand? That is the essence of motif. Yeah. Okay, you said somebody said repeat is under annotation and predicts. Annotation and predicts. He said that's where we find repeats. Probably, I'm just wondering why I couldn't find it. Oh, I've seen it. This is what I've been looking for. Local tandem repeats. All right, local tandem repeats. All right. So I think the plugin can be used for. Okay, so this is the plugin. I hope we're able to. Okay, I think you need to come here and download the Phobos executable location. And then once you download it, you bring it up here. I hope we're able to do that. I don't know. We have a limited time. This is 6 already. So I hope we will be able to get the Phobos downloaded. So we need to download the Phobos. And then once we download the Phobos, we upload the Phobos. I mean, we browse it and pick it from Genius Software, and then we'll be able to actually get it from there. But we have a limited time. That's the software. Okay, so I think probably next time we'll be able to do that. Okay, this is the Phobos plugin. I don't know. I think we've downloaded the Phobos. Okay, this is Phobos. So I'm downloading Phobos plugin. Anyway, this is how you do it though. All these are genetic elements that make sense of your data, of your genes. All right. So let me download. Those of you searching for PB structure or something, you can actually download it from here as well. Okay, it's downloading. So I've downloaded it. I hope I get it right. I hope that's what I need to download. I hope this is it. Yeah, I guess as much. I think that's not it. So that's the plugin that we need to actually install. All right. Plugin for Phobos. Okay. All right. Because of our time, let's not stay much on this because we have other things to do, right? So I'll find my way around that later. Okay. Repeat region with DNA sequence. So predict open reading frame. I already show you how to do open reading frame. So we have a species finder. All right. But regulatory sequence, let me show you how you can get regulatory sequence on DNA sequence. I mean, on Genome software. So you should be able to get regulatory sequence from, this is transcriptomics, transcriptome factors. I think we already have regulatory sequence from this other annotation, right? Okay, this is regulatory sequences. So you do the annotated from, and then you should be able to get regulatory sequence. And I think some of these things for other sequences also have repeats. But I'm very quite surprised why I could not get Phobos for this. All right. So, well, let's leave it. I wish I would be able to show you that, but I will look around that later. Okay. Somebody's asking a question. Don't worry, we are going to round up very soon. Please, assuming that you have identified a resistant pattern among patients, patient was result, and you want to design a drug in silico for correction mutation, how does motif and cloning solve the problem? Okay, the cloning actually solve the problem when you actually have a protein you want to synthesize, you want to replicate through biotechnology. Cloning, so you just use the cloning aspect to be able to bind what you actually want to clone to a vector for cloning sake. Are you getting my point? So in order to reproduce those things in a much more quantity. Are you getting it? I'm talking about in drug design now. But you see, when it comes to in silico cloning, the motif is important because the motif could be the region, it could be a drug target. I repeat, you can actually get motif in a sequence because that region is a drug target. So when you search that, okay, there's this motif that is a potential drug target in E. coli, you can then get that E. coli and come and search through motif on that particular sequence. And then you'll be able to get that particular motif and then extract that region. You understand my point? Maybe more than that region, but that for your drug target when it comes to drug design and discovery. Because that would now potentially be your protein target. Is that clear? Because that region has certain mutations that are responsible for a disease. Is that clear now, Nelson? I hope I'm able to answer your question. Nelson. Is that clear, Nelson? We are through with open reading frame. So you can actually get a function of an open reading frame. Okay, if you want to know the function of open reading frame, like I said, open reading frames are regions that code for a functional protein. I repeat, open reading frame are regions that code for a functional protein. You could actually extract a region. You could actually extract an open reading frame region like this. I've just extracted a region. So I extracted this region, and I can blast this region from here. So I'm blasting against NCBI database to know the function of this particular this thing. I hope I get results. So I'm blasting. I'm online, so it will work. If you are not online, it will not work. So you can actually blast on NCBI database to get sequences. So I'm going to get information on that particular open reading frame I get from the sequence. So open reading frame are regions that code for a functional protein. Now I've extracted that region. I'm going to get more information on that particular region in the gene that I just get the open reading frame from. Is that clear? You can always go to the recorded session to be able to go through the session and practice well. And also, there's possibility that we are going to cut those regions and put them on our website. Are you getting it? And then set the details to you guys on WhatsApp for you to be able to watch on the step-by-step approach on our website. Just go to the recorded session. You should be able to cover. Okay, you should be able to cover. So we have like 60 complete. Okay, so can you see? These are the information that I have just gotten from the open reading frame that I extracted. Can you see? These are the information. Now the description is that this is telling me that I actually get that open reading frame from MECA gene. So it's true. But that particular region is also found on other organisms. Can you see? It's also found on other organisms. And you can see their coding regions. I mean, every genes under these coding regions, it also extracted them for me. Can you see that? It also extracted regions, different kind of regions on that particular region. It's also extracted them for me and gave me the information. So I can decide to click, and this is the sequence. This is the normal sequence. I can come and click it. But don't forget, these particular regions we are seeing, these sequences we are seeing, is because we carry out annotation, genome features annotation from Genome software. All right. So I don't know if we're able to do the rest of the work. Can we do the last part of the analysis? Hello guys, can we do the last part of analysis? Okay. I want to show you can find transcriptome sites, factors as well. You can find transcriptome factors, order, and then maybe search. So it's not working. Those are transcriptome factors. Open reading frame. I showed you open reading frames. I'm trying to see what other things are covered. I think that's it basically. All right. Okay, you can find restriction sites from here. There are 8 restriction sites. Let me see. This is it. These are the restriction sites. Can you see? These are the restriction sites. This is a restriction site. Can you see? Those are restriction sites in the DNA sequence. You can actually go online to get more, to know more about what each of these things are. What is their scientific relevance? What's their scientific significance? By the time you are performing the analysis, you learn the scientific significance, and then you'll be able to apply it to research. That's what it is basically. We'll not be able to cover everything because this bootcamp, but I'm showing you how you can analyze them. So the next thing is, let's look at species finder. Species finder is how you can actually know if a particular, how you can know you can identify an organism. You have a DNA sequence of an organism, but you don't know what organism it belongs to. So you can find the species, the organism, and you just need to go to Center of Genomic Epidemiology. You go to Center of Genomic Epidemiology. Can you see? And then you click it. for Genomic Epidemiology. Center for Genomic Epidemiology. So this is the platform. You get the online tool that you can use to identify species of a particular organism. All right, can you see? This is it. Species finder. You click it, and then you'll be able to utilize it to find a specific species that belongs to an organism. Okay, species finder. The site is taking time to load. There are other tools you can actually explore here on Center of Genomic Epidemiology. There are many tools you can use here for each or different kind of research you want to do. These are online tools, all right? These are online tools. I don't know why this particular tool is taking time to load. Let's hope it works before our time goes. So meanwhile, any question why this is loading? Any question, guys? Do we have any question at all? Any question? I'll be happy to answer you. Yeah, if you are confused about the session, please just come back to the videos later to go through them and learn all over again and practice. No, motifs are not mutant genes, but when you get motifs that has regions of mutations, then they are classified as mutants, but they are not genes. They are just motifs. They are just a very short region that code for a specific, or that's responsible, associated to a specific a region of interest. And the region of interest could be anything relating to evolution, variation, disease, or anything. Are you getting it? Any other question? I don't know. It's taking time. Probably it's my network. I'm not sure. I'm trying to work with my other browser and see, probably it's because of my browser. Please drop your questions. Okay, I hope this will work. Species finder. Any more question? Okay, so the motif, the species finder has worked. So you can upload your assembled genome. Now the data, the whole genome sequence of what's it called right now, of Escherichia coli that we downloaded yesterday, which is this one, is actually called assembled genome because it has already been assembled. So imagine you don't know that this is Escherichia coli, but you have the genome. Are you getting my point? You can actually search for it here. And how do you do that? You come here, all right? You just choose the genome. You choose the genome, all right? You come here, download. So this is the FASTA file. You pick the FASTA file. Okay, I've uploaded the FASTA file. I need to click upload. So it's going to take time to upload. By the time it's done, it tells us the species of that particular file that we have uploaded. Are you with me? It tells us the species right away. It tells us the species right away. So now it said, unfortunately, your job failed. Why? It could actually be due to because of I'm trying to, maybe I should get for the config file. So let's try this. I don't know why it failed, but if it also failed here, it means probably the online tool has an issue. Most times, some of these tools usually have an issue. All right. So let's see. It's going to redirect. All right. So my job is processing. Now my job is processing. I think there's an issue with that particular genome I downloaded. So not some of the genome could fail, but I hope this works. So, but it could take time. All right. This job could take time. So, and then you can impute your email. So by the time the analysis is done, it notifies you in your email. But the analysis could take hours, could take 2 hours before you get it. So we can't actually wait, but you can try it on your own end. All right. So once you try it on your own end, you should be able to get the results. Is that clear? You should be able to get the results. Download as many genomes as possible and upload them. Is that clear? So, and if it doesn't work, you can download the assembled data. You can have an assembled data and upload it. That way you are sure of. Now, the essence of this species finder is when you do culturing and you get whole genome sequence of your bacteria. For you to be sure what that bacteria is, you can use species finder. Do you understand? After when you are doing molecular characterization, you can upload your genome here right now. After you have assembled it, you have processed it, in order to be able to identify the species of that particular genome, to be sure that, okay, what you culture in the lab is actually what you have sequenced. So that is the essence. All right, so that is it. I hope the session has been amazing. So certificate will be ready. Certificate will be ready. Please, I don't need to repeat how to use Center of Genomic Ethnology, Mubin. Just try to go through the recorded session. After this session, the recording, the YouTube session recording will be available there. Okay? So that's it. It's definitely will be there. So the certificate will be ready. Message will be sent across and also via email on those who are interested in the certificate. So, and then don't forget Genome software, a lifetime activation key of Genome software also comes together with certificate. And this Genome software worth over $5,000. So, but we're going to be giving you for free as far as you are getting the certificate. $3 cover for administrative cost. Okay. The bootcamp is definitely fully funded. Okay. Okay, I think it works. We have our results, species finder. I think the reason why this does not take much time was because of what's it called right now? The sample was not too large. Can you see the species? Daucus carotas species sativus. That is the name of this particular sample. All right, the species is what? Daucus carotas or species sativus. Can you see? I uploaded a sample that I really don't know the sample. I don't know the organism it belongs, but because I submit to species finder, it finds the species for me. Can you see that? This is the result. So you can submit your sample to, then whatever matches the sample, maybe at 70% matching, then it gives you, right? It gives you the species it belongs to, that particular genome sequence belongs to. So that's it basically. So I'm also happy to inform all of you that we're going to be having free research internship program that we run for 9 to 10 weeks. All right, that we run between 9 and 10 weeks. All right, and it's starting next month. Free research internship program from basic to advanced level of bioinformatics. All right, and genomics research. So because it's going to be fully funded research based, but there's going to be an application fee of just $30 for those who are interested. The $30 is going for certification. It's going for not certification. It's going for publication rather, because after the research project, we are going to have research papers, and then we need to publish this research paper with you as co-author. All right, so you get access to advanced skill set, advanced knowledge in genomics and bioinformatics, more research experience. You're going to be given tasks. You're going to be taught what to do, how to do it. You're going to perform those tasks. All these tasks are going to be put together as a group, because you're going to belong to a group. So all tasks of each individual is going to be put together to make a research project, a research paper. And then the research paper will publish with you guys as co-authors in the paper. Is that clear? Is that clear? So if in case you are interested in the fully funded research internship program, it's available. We just run the code 3.0A. Now we are going to be running 3.0B. All right, we're going to be running 3.0B in anti-cancer, antibacterial therapeutic research and development, anti-fungi, anti-malaria, antiviral therapeutic research and development. We're also going to be doing infectious disease research in different infectious human pathogens of bacteria, right? That are bacteria. So just in case you're interested in the research internship program, you let us know. We're going to share the link also on WhatsApp groups, okay, for the free research internship program. But don't forget there's a commitment fee that must be paid. It's compulsory, all right? And please, you need to be selected for the internship program. So you can apply. If you got selected, then you are in. I think we currently the 3.0A, they all have mentors. They're going to be working with our automated research platform called Omics Board and all of that, okay? So for your certificate, we're going to send information, and also keep an eye out for the research internship program tonight. We're going to send it out. Hopefully you get selected, okay, for the research internship program. Thank you so much. Any other questions? Any other question? You can ask your question before we round up, okay? We're about to round up. We're about to come to the end of the boot camp. We're about to come to the end of the boot camp. It's been an amazing 2 hours, about 2 hours, 30 minutes. Okay? It's really an amazing session. And Genomic Hub, thank every one of you for being available for this boot camp. We'll continue doing impact and bring more impact and make sure that we train as many people as are interested in genomics and bioinformatics out there across the continent. We'll be able to train you and bring you to a place of competence. So thank you guys for joining us at this amazing webinar session. It's really amazing. Any question, just kindly drop your question now so that I can answer it, and then we can come to the end of the bootcamp. Meanwhile, information on the information on how to get the certificate will be sent across you guys. Okay, I'm still waiting for questions. Okay, I'm waiting for questions. Yeah, the internship is online. It's an online internship. We're going to be sending the information across WhatsApp groups. Okay, we're going to be sending information across WhatsApp group. Meanwhile, let me send the link quickly right now. I've just sent the link. I've sent the link, all right? I've sent the link. The internship is online and it's an international internship, okay? It's an international internship. The internship is an international internship program. So you're going to be working with different peers from all over the countries, all right? So the last internship program, we got about 80 countries. And on the website, you see presentation of research projects from our previous cohort 1 and 2. Some of their papers are currently being worked on to publish right now. So just to extend a hand of impact, the more to you guys, and that is why we are trying to share this cohort 3.0B. We've done cohort 3.01, and they've started already. So we're taking cohort 3.0B. The internship is to start next month, latest on the 12th of next month. But the application is going to be sent across all WhatsApp group and email. So just keep an eye out so that you can have a registration. It's also going to be shared on our LinkedIn. If you have not joined us on LinkedIn, make sure you do that. Primers, somebody's asking, are primers designed through Amusa are highly specific, or they can also give us non-specific product in PCL? They could, but if you want to be specific or non-specific, the best is to work with Genius software. That way you'll be able to know the region you're trying to work with. All right. Okay, you want to have plant science, plant genomics. Okay, we'll look into it if at all we want to have a plant genomics and decide if we should have it. So just make sure you try to apply. Okay, so that's it basically. Yes. Details on how to get certificate will be on WhatsApp. I hope you're on WhatsApp. Right? I hope you're on WhatsApp at Elijah, I hope you're on WhatsApp. So we're going to set details on how to get a certificate on WhatsApp. Yes. Yeah, we'll send confirmation email to everyone, including how to get your certificates. And those who have made payment of $3 will get their certificate. Yes, $30 is just for the publication, the cover for publications, and also encouragement, just to encourage our mentors as well, because you're going to be having mentors once a week, meetings, mentorship with our mentors, and you're going to be carrying out your research on our automated research platform called Omics Board. Yeah, please, if you have more questions, just reach out to the admin. Okay. Oh, we cannot provide hard copy certificates. We can set soft copy, but you can go and download it. Okay. Okay, how does the selection process? So we use within a week or two. So we are going to send those who have been selected. We already have a form, so you just need to fill the form, and we walk through whatever you fill to select you for the internship program. Yes, please, you can add this bootcamp to your CV, including the certificate, okay? It works, okay? Okay, you are looking for open reading frame, okay, Rice? You are looking for open reading frame in a sequence because you want to identify regions that code for a functional protein in that particular region, in that particular sequence. You know, the region that does not code for a functional protein is called introns. They are non-coding regions. But the region that code for a functional protein, by identifying open reading frames, it means that region code for a gene. They code for a functional protein. They are exons. They are exons. They are mRNA. So they code for a functional protein. Do you understand, Race? Yes, our certificate is recognized because our institute is registered in the US. It's a US-based registration. Genomic Institute Incorporated is incorporated in the US. Are you getting me? It is incorporated in the US, and we have been in existence for the past 2 years. So, and we are globally recognized. We train over 20,000 students. So definitely, wherever you take a certificate to, it will be recognized, because we are an international based company. By the grace of God, we've been able to reach out to we've been able to have influence or impact individuals in over 120 countries worldwide. If you have many species displayed, you could actually pick the best based on the confident results. My confident result here failed. Even if yours failed, you could actually decide to go with the first one. Do you understand? Yes, please. The soft copy of certificate is available. Once you make your payment of $3, you get your certificate with the Geno software, the lifetime activation key of Geno software. Okay. Yeah, introns are the need for introns that are just there to at the DNA level to put a balance to the sequence, especially in the temperature region, like in different temperature room, to put a balance when it comes to the sequence length and the operations of those genes at the nucleus, right? But you see, before they can be translated to protein, they need to be first of all spliced out at the mRNA, at the RNA region, so that they can become mRNA. Do you understand my point? So, but the reason for introns is just to create balance within the sequence, because any mutation, do you know, even introns, if mutation happen in introns, it could actually affect the whole functionality of that particular gene. The reason is because there could be frame shift mutation, multiple variations as the result of that particular variations at even introns. So the condition or the sequence pattern of DNA sequence at DNA level must be in order. That's why we have introns. Any more questions? We're about to round up. Yes, please. The GC content is automatically calculated for the primer design. Thank you. Any more questions? What is genomic DNA? Well, DNA is a nucleotide basis, all right, that code a particular gene or genome of an organism. You know, you as an individual, you have. I will say, please, Faith, can you watch the video of the first session of the first day? We explain everything what DNA is. It's very, very detailed. You can watch it, okay? To help out. No. We don't identify motif from blast, okay? We identify open region frame from blast, okay? Isaac MJ. Sorry, Martha, we may not be able to provide a soft copy manual. That's why we provided the recorded session, okay? So you can go to the recorded session. Thank you. If your sequence is not found, there could be maybe some issues, all right? There could be some issues at maybe the composition of that particular genome sequence, all right? If it is not found, okay, or maybe the sequencing level. Okay, so Felix, please, I said answer to your genomic DNA. What is genomic DNA? Please watch the yesterday session on YouTube, okay? Yesterday session, kindly watch it on YouTube. You have full details on what DNA sequence is. All right, Faith Samuel. How we amplify DNA sequence after designing the primers. You use PCR, okay? You use PCR machine, and you impute those those primers into them. And, you know, it's just a PCR work. You need to learn how PCR works, okay? Okay, you can drop phylogenetic tree on NCBI, okay, if you want. All right, Ayomide. But 2 sequences is not enough for phylogenetic tree. You need more sequences. Maybe 8, 10, 12, 20, 25 upward. All right, so we're going to be rounding up here. So thank you guys for joining us, for joining this amazing bootcamp session. We really appreciate you. All right. My battery is low now. I need to go. All right. So please, the link to join the bootcamp, to register for the bootcamp will be sent to you guys on YouTube. To register for the internship will be sent to you guys. Okay, very shortly. So just kindly stay while we do that. Stay on the group while we do that. We'll send it to your email as well. Thank you guys for joining. We really appreciate. Once again, my name is Oluwaseyi Olawale. We hope to see you guys in our next programs, our next impactful programs. Stay with us. Tell people more about us and how amazing they can actually learn from us. So thank you. for joining us once again. Have a great and wonderful time. Thank you.